Hello again, dear student. Today we are going to talk, or you already talk about the endocrine system labs. All right. Today we are going to focus on the next lab, which is the blood lab. So let's get started. And before we continue, imagine yourself that you are in the lab. So to deal with once you are in the lab, automatically you have to wear goggles, gloves, lab coat, no open shoes. And today we are going to work with the blood. So for sure we have to use biosafety measures like biosafety trash that they are read in the lab, etc. All the safety possible so we can work with the blood. So let's get started. All right, let me share it. So yeah, once you collect the blood from your little finger, you put it in a tube and you centrifuge that. And once you get the centrifuge tube, you centrifuge it like for 5 minutes. And you can determine the composition of your blood. 55% represent the plasma, and 45% represent the formed elements. 55% of the plasma, 90% of it is only water, and the rest is three types of protein: albumin, fibrinogen, globin, and some electrolyte like sodium, potassium, calcium, magnesium, chloride, bicarbonate. So yeah, and the 45% of the form element in your blood is composed of three types of cells. We have the red blood cells, which are we call them also erythrocyte, site for cell, and also we have the white blood cells. We call those leukocytes. There are 5 of them. We will come back to it later. And platelets are little, they are the teeny fragments, cytoplasmic fragment. So they are the 3 types of the 45% formed elements of the blood. So the red cells or erythrocyte cells, they don't have a nucleus. They look like whole donuts, and they are biconcave. They transport their principal function. They transport oxygen and carbon CO2. They are form of the molecule that is the hemoglobin. Very important. So this is why when we are as any doctor, the first thing he do, he look at the composition of your red blood cells. Because the hemoglobin, and they look at also the composition of the hemoglobin, because that's the one that brings oxygen in your body. Leukocyte, which is the white blood cells, we have they are they have nucleus, they have nucleated cells, and they are usually not biconcave. Biconcave are the red cells. They are spherical form. We have 5 of them. We have cells, 3 of them have granulation inside. You will see them under microscope, very amazing, very nicely. Granulation inside, we call that granulocyte. There are 3 of them. We will come back to it. And we have 1 that they are smooth. They don't have granulations in their cytoplasm. This is agranulocyte. There are 2 of them, monocyte and lymphocyte. So the granulated one, or the granulocyte cells, we have the neutrophil. here is the thing. The way we are going to differentiate all these 5 white blood cells is by the form of their nucleus and also by the presence of granulations. When they are granulocytes, we are going to look at the form of their nucleus. When the nucleus is multi-lobed, this is neutrophils. Their function, all the white cells, their function is to defend your body. All right. So the function of the neutrophil is to phagocytize pathogens. Eosinophils, their nucleus is B-lobed, 2-lobe. If you see it, they are like 2 lobes over here, 1 and 2. B-lobe, they kill parasites, and they are also a role in allergy and asthma. Eosinophil, basophil, they release histamine, and the nucleus is lobed nucleus, and they release histamine, and they play a role also on anticoagulant function because they contain heparin. So the next is the non-granulocyte cells, which is very smooth. If you look at their cytoplasm, they don't have granulation, they don't have those inside. Nucleus is very spherical. The lymphocytes are the smallest of all those cells. Of all the form element cells, the lymphocytes are the smallest one. Of all the white cells, they are the smallest one. They play a role on the immune response, and they either attack the cell during the immune response, either they attack the cell directly or via productions of antibodies. We will look at that. We will talk about them when we study the immune response, when we study the lymphocyte B and T. Monocyte, their nucleus have a form of U or kidney. All right. They are also agranulocytes. Their nucleus is form a U or a kidney form, a kidney shape, and they play a role in the tissue. They develop into macrophage that will phagocytize, they will engulf pathogen or debris. Platelet cells, platelet blood cells, they are very tiny. They are cytoplasmic fragments, and they play a role in blood clotting. All right. So how we can see the histology of the blood? If we are in the lab, and if you want to do it, if you have microscope and slide and cover slip at home, you can do it, and you can look at your own blood smear at home. But first you have to wear gloves and take all the precaution possible. So the safety for the lab, the safety, we went through the safety during 1406 and during 2401. And also any times you are entering the lab, this is wearing goggles, wearing gloves, wearing a lab coat, and no open shoes. And you have to take care of where the trash, where we put the glasses, specific glass trash, where we put anything biohazard, it's in red trash, and anything that is like cover slip, they have specific trash for it, etc. You have all the time to put in mind that you are in the lab and you are going to follow the protocol of this lab for your own safety and the safety of the other and your loved one. So you take a smear, you take a drop of a blood, and you put it on a slide, and you very gently, you take a cover slip and you go tangentially and to push this blood cover completely at the size of the cover slip. So you obtain a smear, and when you look at the smear under the microscope, this is what you are seeing. You are going to see different type of leukocytes as well as the erythrocyte. The erythrocyte, that is the red cells that you are seeing over here. And you are seeing also the white cells. And look at this white cells. This is a monocyte, U form. You remember? Kidney shape. You have multi-lobe, that's neutrophil. You also eosinophil. If you remember, Eosinophil is beloved. Look at how we look like, all right? Beloved. So look at it. If we have exactly this form, this is eosinophil. It's beloved. 2 lobes of the nucleus, okay? And the lymphocyte is the smallest white cells that is there, and it's smooth. There are no granulation. All of them, all those have granulations. These have granulation. These 2 are smooth. They don't have granulation inside, inside of their cells. All right. So this is multilobe neutrophil, surrounded by lots of erythrocytes. Multilobe. Again, in the microscopy in your smear, you are going to be able to localize those granulocyte eosinophil beloved nucleus. And you can see very, very nicely those granules in the cytoplasm that is red by staining with the iodine, and the nucleus is blue by staining by the methylene blue, used as contrast stain. You have the basophil, which is the beloved nucleus, and the cytoplasm also is granulated. Lymphocyte, very smooth, no granulations, and it's one of the smallest white cells. And the nucleus is a spheral nucleus. It occupied almost all the cytoplasm of the cell. You have the U-form or kidney shape, the monocyte. So you are going to be able there in the practical, when I give you any histology microscopic picture, you will be able to recognize which type of cells is this. The first thing when you go see a doctor, if he suspects that you have an anemia or something's going on, he will do an hematocrit reading. This is very easy. Anybody can do it. You can, even if you have those instruments, you can do it at home. I put here a video to show you, if you want to see it more deeply, how to use the hematocrit reader. But all what you need is an hematocrit reader, centrifuge, that is built for hematocrit centrifuge. And hematocrit cells, this hematocrit cell are able to hold or to carry an hematocrit capillary tube. All right, and all we have to do is just to pinch yourself, put the little hematocrit capillary, and the blood will come over there, of course, into the capillary, and you put it in the hematocrit holder for the capillary. Usually we use like a 3 or 2 of them, and you put them in the centrifuge. And the way, if I look at it there, I am going to zoom so you can see it with size. If you look at it, this is the size with the blood, the hematocrit. You have to face outside, not inside. If you put it in the opposite way, it will drop the blood inside of the centrifuge once it's centrifuge. This is a special centrifuge for those hematocrit holding capillaries materials. So, and after like 5 minutes, and the centrifuge, it's a special, the force for it, it's a special to centrifuge those little capillaries where your little blood is on it. And after that, I mean, we can do a reading using an hematocrit reader, which is, I go back to my slide, this one, that's my hematocrit reader. And the way you post it in, and you can read, if I see it very well over here. And you can, in the hematocrit reader, when you put your centrifuge capillary you can read that you have, and you put it in, and you can read exactly how much percentage of red blood cells you have. If you look at it over here, you can read that this is 45% of the blood cells. The rest is the plasma. So this is for the hematocrit. So the hematocrit is a little materials, very easy, that will help to measure the percentage of the red blood cells in our blood. So normal blood cells, if the hematocrit show that we have between 37 to 47 for female, and for men 42 to 52%. So in general, we need within 45%. When it's less than 37%, when it is less around like 30%, this is the hematocrit show that this person suffer from anemia. When it's very high, this is polycreatemia that's elevated, that's abnormal. And also staying with the red blood cells, we can also determine hemoglobin using an immunoglobinometer. So a drop of blood again to the plate of the blood chamber. It's typical to be read by this hemoglobinometer. The blood sample is hemolyzed with the wooden hemolysis applicator, and this requires 30 to 45 seconds. And then after that, the charged blood chamber, it's inserted into the slot on the side of the hemoglobinometer. And we have to look for the green split screen that are found by moving the slide with the index, with the right index finger. When the two color match in density, the gram per 100 millimeter represent the percentage of hemoglobin that are read in the scale. So this is also very easy way to determine the percentage of hemoglobin in your red blood cells. For the coagulations, I'm not going to talk about it here because I explain it very, very well in the lecture. Also the typing, when I give you a sample of a blood, you are going to be able to determine which blood type is it. For example, you have to know that type A blood mean that you have antigen A on their surface and the antibody anti-B circulating in their plasma, and type B blood mean that they have antigen B on their surface, on their membrane surface, and they have an anti-A antibody in their plasma. Type A and B contain both antigen A and B on their surface, and therefore they don't have antibodies anti-A and anti-B in their plasma. The O type of the blood does not have neither antigen A, neither antigen B, and therefore they do have anti-A and anti-B antibodies in their plasma. So I will give you a test, like for example, okay, I have a sample blood and they put in contact with an anti-A and they show clumping or agglutinations. That's automatically telling me that this is a group A type blood, and when I put it with an antibody anti-B, there are no agglutinations or no clumping. So this is a proof that this is type A blood. And in contact of an anti-D, which is an anti-rhesus, it's clump, which is mean that it is a rhesus positive. So all this, I wanted to train it with you. Either I will say the word clumping or agglutinations, or I will just show you pictures of agglutinations, which mean this is agglutinations. This is clamping. This is non-clamping. Agglutinations versus non-agglutinations. Okay? So this is we stop here for our lab-related blood. They have all the time at the end of any of my lectures or my lab lectures, I have some questions that they help you to study. Before you move on to the next lab, you have to answer to those questions. If you are unable and you cannot find the answer, that means you did not read the lab or the lecture correctly. Go back until you got the answers. If you have any problems, you can always email me, and we can email me, and they will always reply to your emails. Thank you so much, and have a blessed day.